In the closed complex, the promoter DNA is still fully double-stranded. 5. RNA polymerase, assisted by one or more general transcription factors, then unwinds approximately 14 base pairs of DNA to form an RNA polymerase promoter open complex. In the open complex, the promoter DNA is partly unwound and single-stranded. The exposed, single-stranded DNA is referred to as the transcription bubble. 5. RNA polymerase, assisted by one or more general transcription factors, then selects a transcription start site in the transcription bubble, binds to an initiating NTP and an extending NTP, or a short RNA primer and an extending NTP, complementary to the transcription start site sequence, and catalyzes bond formation to yield an initial RNA product. 5. In molecular biology and genetics, translation is the process in which ribosomes in the cytoplasm synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. The entire process is called gene expression. In translation, messenger RNA, mRNA, is decoded in a ribosome to produce a specific amino acid chain, or polypeptide. The polypeptide later folds into an active protein, and performs its functions in the cell. The ribosome facilitates decoding, by inducing the binding of complementary RNA and codon sequences to mRNA codons. The RNAs carry specific amino acids, that are chained together into a polypeptide as the mRNA passes through, and is read by the ribosome. Translation proceeds in 3 phases. Initiation. The ribosome assembles around the target RNA. The first RNA is attached at the start codon. Elongation. The RNA transfers an amino acid to the RNA corresponding to the next codon. The ribosome then moves, translocates, to the next RNA codon to continue the process, creating an amino acid chain. The 3 phases of translation: initiation, polymerase binds to the DNA strand and moves along until the small ribosomal subunit binds to the DNA. Elongation is initiated when the large subunit attaches, and termination ends the process of elongation. Termination. When a stop codon is reached, the ribosome folds the polypeptide into its final structure. In prokaryotes, bacteria, translation occurs in the cytosol. 1. Where the medium and small subunits of the ribosome bind to the RNA. In eukaryotes, translation occurs in the cytosol, or across the membrane of the endoplasmic reticulum in a process called co-translational translocation. In co-translational translocation, the entire ribosome/RNA complex binds to the outer membrane of the rough endoplasmic reticulum, and a new protein is synthesized and released into the ER. The newly created polypeptide can be stored inside the ER for future vesicle transport and secretion outside the cell, or immediately secreted. Many types of transcribed RNA, such as transfer RNA, ribosomal RNA, and small nuclear RNA, do not undergo translation into proteins. A number of antibiotics act by inhibiting translation. These include clindamycin, anisomycin, cyclohexamide, chloramphenicol, tetracycline, streptomycin, erythromycin, and pyrimycin. Prokaryotic ribosomes have a different structure from that of eukaryotic ribosomes, and thus antibiotics can specifically target bacterial infections without any harm to a eukaryotic host cells. The basic process of translation is the addition of one amino acid at a time to the end of the polypeptide being formed. This process takes place inside the ribosome. A ribosome is made up of 2 subunits, a small 40S subunit and a large 60S subunit. These subunits come together before translation of mRNA into a protein to provide a location for translation to be carried out, and a polypeptide to be produced. 2. The choice of amino acid type to be added is determined by the genetic code on the mRNA molecule. Each amino acid added is matched to a 3 nucleotide subsequence of the mRNA. For each such triplet possible, the corresponding amino acid is accepted. The successive amino acids added to the chain are matched to successive nucleotide triplets in the mRNA. In this way, the sequence of nucleotides in the template mRNA chain determines the sequence of amino acids in the generated polypeptide. 3. Addition of an amino acid occurs at the N-terminus of the peptide, and thus translation is said to be carboxyl 2-amino directed. 4. The mRNA carries genetic information encoded as a DNA sequence from the chromosomes to the nucleolus. The ribonucleotides are read by translational machinery in a sequence of nucleotide triplets called codons. Each of those triplets codes for a specific amino acid.